Hello, my name is Ed Chapman, and this video cast is the second video cast in our photosynthesis series. And the first thing we're going to do is we're going to label some parts of a chloroplast and a mitochondrion. And I'll give you a handout in class so you can write this information down. First thing I want you guys to do is to label the location of the electron transport chains. Now, we've already talked about the electron transport chains in mitochondria. It's located on the cristae, which are these folded membranes. Now, in the chloroplast, they also have electron transport chains. They're going to be doing the exact same thing that they were doing in respiration, but in the case of a chloroplast, they're located on the thylakoids, which are these individual layers inside of this larger structure called a grana. Alright, now chloroplasts and mitochondria are both surrounded by an outer membrane, and of course we've talked about the inner membrane being folded to increase its surface area. If we label the actual regions, the innermost part of a chloroplast is called the thylakoid space, and the outer filling is called the stroma. In the case of a mitochondrion, the innermost part is called the matrix, and the outer filling is called the intermembrane space. Grana are these stacks of thylakoids, and the cristae are the folds in the mitochondrion. So altogether here you have the anatomy of these organelles we call chloroplasts and mitochondria, which are the powerhouses, so to speak, of eukaryote cells. Now if you turn your diagram over on the back, there's two more pictures, and we can track some molecules moving here. And as you can see, the chloroplast is producing glucose because it's taking in carbon dioxide and water molecules. It's also producing oxygen gas, and the oxygen, of course, is taken in by the mitochondrion, which then produces carbon dioxide and water. Inside of the matrix of the mitochondrion is where the Krebs cycle is taking place, and remember, the Krebs cycle uses pyruvate, which comes from the cytoplasm because glycolysis is breaking glucose down into pyruvate. So altogether you can see how these molecules, especially carbon and oxygen, are cycling between chloroplasts and mitochondria in the metabolism of living cells. Alright, now the thylakoid membrane contains photosystems, and these photosystems are chlorophyll molecules that are anchored in the membrane, and their job is to take low energy electrons and boost them to a higher energy level. The chlorophyll molecule looks kind of like a steroid molecule. It's got a tail region, and it's got a bigger head kind of area. Now the anchoring tail is what holds the chlorophyll molecule in the membrane, much like a phospholipid tail anchors a phospholipid in the bilayer. It's also got a magnesium atom here in the middle surrounded by a ring-like cage of other atoms, mainly carbons and hydrogens and oxygens. And this ring-like cage holds the magnesium ion in a way so that it can kick electrons out. And we'll see that in just a second. Now chlorophyll responds to light, as all pigments do, by absorbing some of the energy and bumping electrons from a low energy level to a higher energy level. And if nothing catches these electrons in their excited state, they then fall back down to where they started from and release this energy again. Now remember, energy is conserved, but some of the energy is lost as heat, and some of the energy comes out as a photon of a longer wavelength. Okay? So you can see this happening if you take a beaker of chlorophyll that's been treated in the right way. You can shine bright light on it, and it will produce a bunch of excited electrons, and then when you turn the light off, these electrons will fall back down to where they started from in the chlorophyll molecule and release some reddish wavelength light, which causes the chlorophyll to glow red, which is kind of the color you wouldn't expect to see from a green plant. Chlorophyll glowing red, kind of a weird, creepy, bloody red color. Just in time for Halloween. Alright, now the photosystems, their job is to funnel energy from light to a reaction center chlorophyll. Now this reaction center chlorophyll is the key chlorophyll molecule that's going to be kicking an electron out to an excited electron, an excited energy state, and that excited electron is going to be captured by the first electron acceptor. So the job of all these chlorophyll molecules and other pigment molecules is to focus the energy of the light to the reaction center chlorophyll. Now, the part of photosynthesis that requires light is sometimes referred to as the light-dependent reactions, which is the same thing as these photosystems. And there are two photosystems. They're known as 1 and 2. And they require energy from sunlight to make ATP out of ADP and to capture electrons from the chlorophyll in the form of NADPH, which is the electron carrier that's been reduced because it's picked up electrons. It starts out as NADP and it becomes reduced to NADPH, a lot like the NAD and FAD we saw in respiration. Now here's a picture of the photosystems working together. I know this is a little intimidating when we first look at it, but oops, go back here. But what we've got going on is we've got a photosystem 2 and a photosystem 1 next to each other. Photosystem 2 is connected to photosystem 1 by an electron transport chain. All right, remember all of this is in the membrane of the thylakoids. And we've got sunlight energy shining on the photosystem over here, and we've got more light shining on photosystem 1 over here. So they're both receiving energy from sunlight. Now let's start our analysis by looking what's happening over here. The photons of energy hitting the primary chlorophyll molecule here absorbs sunlight energy of 680 nanometers in length. And when the right wavelength of sunlight hits this chlorophyll molecule, it's able to kick out a pair of electrons to a higher energy level. Now they don't fall back down like we saw with fluorescence, but they're captured by the first electron acceptor of the electron transport chain. And then they're allowed to fall back downhill gradually and produce ATP in the process by chemiosmosis. Now these electrons then end up in photosystem 1, where they're kicked up to a higher energy level again. They pass their way down another shorter electron transport chain until finally they become the property of NADP, which is reduced to NADPH. So the electrons start over here, bounce up, down, up again, and then into NADPH, which holds on to them. Now, if we're taking electrons out of photosystem 2, they have to be replaced from somewhere, and those are coming from the splitting of water, which we talked about earlier. A water molecule is split in a way that produces hydrogen ions and free electrons, which go into this chlorophyll molecule and replace the ones that are traveling through this pathway to NADPH. Now NADPH captures these electrons and is reduced to NADPH, as you can see right here. Okay, so we've captured some electrons. We're going to be using them later on. Okay, ATP is also made, and ATP is made in just the way we saw in respiration, as electrons fall through an electron transport chain, not towards oxygen, but in this case towards photosystem 1. And as they pass through the electron transport chain, their energy is used to phosphorylate ADP into ATP. This is called non-cyclic photophosphorylation because the electrons are leaving one place and ending up somewhere else completely different. We're going to stop there and we'll pick up with photosynthesis videocast number 3.